we recruited these 500,000 men and women aged 40 to 69. The age range was somewhat arbitrary, young enough to get people before disease has developed in the main, so that disease won't have changed their non-genetic risk factors, but old enough that they will develop a wide range of diseases within the first 10 years or so that we can actually study the determinants of disease, but we will continue to follow them until the bitter end. We have general consent for all types of medical and other health-related research by both academic and commercial researchers. And I just wanted to make the point here that with respect to IP, UK Biobank does not retain IP on research using the resource. That belongs to the researcher. We have consent for follow-up through all medical and other health-related records within the UK. People sometimes ask, why did we do a study with 500,000 people? To which the answer was we didn't have enough money for 1 million. And I mean, it's not a joke answer really, because when you kind of think about the scale, in a prospective cohort, only a small proportion of the individuals will be informative for any particular disease. It's a very cost-effective design if you're interested in lots of different diseases and lots of different exposures, because everybody will be informative for one or more disease, but only a small proportion will be informative for a particular condition. So you need large numbers at the beginning to have large enough numbers of individuals who will develop whatever disease you're interested in. And this is illustrated here by randomly selecting from a prospective or meta-analysis of prospective cohorts, which included about 1 million individuals with 12 years follow-up. And I asked the investigators leading this if they could randomly select 5,000 individuals and look at the relevance of 2 strong risk factors with a common condition. So the 2 strong risk factors were blood pressure and age, and the risk of death from coronary heart disease mortality. So here's coronary heart disease mortality on a doubling or log scale versus blood pressure, with 12 years of follow-up, looking at the association at different ages. And these are the confidence intervals around the points. And what you can see is a kind of general blur of higher blood pressure, higher risk. Now, these are the data that your doctor uses to decide whether or not you should get blood pressure lowering treatment. Because this is the size of the Framingham Heart Study, 5,000 people. And that's how they develop risk scores to decide whether or not you should take a blood pressure lowering treatment at a particular age and a particular blood pressure level. Not very reassuring. If you go to 50,000 people, things start to come into focus. You can at least discriminate between the risks, the absolute risks at different age. You can see more clearly this upward slope. But at younger ages, is there a J-shaped curve below which lowering blood pressure increases risk? At older age, is it flattening such that high blood pressure is not associated with increased risk? And this was believed for a long time in the hypertension field that blood pressure was not an important determinant of increased risk of stroke and coronary heart disease in the elderly, and therefore lowering it wasn't a good idea, based on these kinds of data. Take it up to 500,000 and it's like a microscope. The whole thing comes into focus. You see these beautiful log linear associations throughout the range. You can start to look at interactions. So by plotting it on a doubling scale, a steeper slope indicates an interaction on a relative scale. So stronger association in relative terms at younger age than older age. But of course, at older age, you have higher absolute risk. So in absolute terms, the interaction goes the other way. Bigger absolute difference per millimeter of mercury, higher blood pressure at older age. Now, this is a major risk factor, age, another major risk factor, blood pressure, and a common condition. So if we're thinking about the relative risks that we see with genetics and many other risk factors, you can see why I argue that 500,000 is not enough. But at least it's what we've got. As was mentioned, we've got a very wide range of baseline questions. What we did was we got experts from lots of different disease disciplines, interested in different conditions, interested in different risk factors, to tell us what questions, what measurements, and what samples to collect. So someone asked a question about how much data is there. There are thousands of different data points on each individual. And if you go onto the UK website, for each measure, for each question, you can see the distribution. You can see what question or measure was made, and you can see for each of the variables what the distribution is. And you can just go onto the website and see that. And you can also find out what assays have been done. We then looked to see whether we could enhance either in large subsets of the participants or enhance by doing sample assays from all participants, and that has been mentioned. So the genotyping SNP has been described, so this 820K bespoke chip. I think it's worth stressing that the DNA extraction, which was coordinated by Sam Murphy in the UK Biobank Coordinating Centre, she had people working 24 hours a day. Seriously, we had shifts working 24 hours a day, 7 days a week, extracting the DNA and then shipping it over to California for genotyping, where they were probably even working even longer than that. This was all completed in 18 months, from starting DNA extraction to the genotyping being completed, which I think is when you saw the quality of the data that was described in the previous two talks, to have done that in 18 months is amazing. We released around the end of May, June, the first 150,000, and as Peter said, the plan is to release all 500,000 in re-imputed data by mid-2016. I wanted to have a little different spin on the quality control check that Peter described. So this is just this kind of quality control check for height on chromosome 4. And height is quite interesting because there have been meta-analyses of GWAS data on about the same scale, on about 150,000 people. So you might think it's a kind of like-for-like comparison. And as was pointed out, these are known hits that are confirmed in UK Biobank. I think almost more interestingly are the known hits that are not confirmed by UK Biobank. Are they false known hits? I just think what we've got here is a single array done at one time on a very well characterized population, rather than a meta-analysis. It may indicate the increased power of that kind of resource compared with combining data from different studies. In addition, of course, as Neil pointed out, we have all of this other information which was systematically obtained on all of these individuals, which is, from having worked in these kinds of meta-analysis, is not available. So I think the kind of power to really explore these associations is far greater than just the numbers of individuals. It was mentioned that we're currently running a set of biomarkers, and we just did the hematological assays as the samples were being collected. So hemoglobin, red cell counts, white cell counts, etc., because you can only do that on fresh blood. We deliberately deferred doing any other assays, partly because we didn't have the money, but partly because we could improve on the quality control. So both with respect to the extraction of the buffy coats for DNA extraction, and even more importantly for these assays, the samples were extracted from the freezers in a random sequence. And so there is no systematic bias in the sequence of analysis of the samples. And I think that's particularly important for well, I felt it was particularly important for biochemistry. I'm starting to believe it may also be important for the genetics, because it's not that we're assaying the people who have high cholesterol, who were collected from Scotland and the north of England, first, and then doing the ones from the kind of the wealthy cabbage salad-eating south. Later. They're being analysed in a random sequence, and so we have a kind of built-in quality control. We know that nothing should change over time, and therefore we can, if we see drift, then we can very rapidly deal with it and ensure that we don't introduce systematic biases. So we're doing a whole series of assays related to cardiovascular and cancer markers, diabetes markers, renal markers, etc. Because then these data will be, like the genetic data, readily available for researchers to use. And those data will start to be released during the first quarter of 2016. We are also imaging participants. We've just completed the imaging of 6,000 participants. That makes it one of the biggest imaging studies ever done. And not only is it bigger than many other studies, but it has multimodal imaging. So many studies are focused on, say, brain imaging or cardiac imaging. What we've got, though, is imaging of lots of different parts of the body. So we have a 3 Tesla magnet imaging of the brain, and a 1.5 Tesla magnet of the heart and abdomen. Low power radiation for bone, joints, and body, and you can get body composition automatically out of this. Ultrasound of the carotid artery to look at atherosclerosis. A repeat of the baseline questions and measurements with additional information about cognition, a 12-lead ECG, and the biological samples being collected again. So we're about to release, during this month, the imaging data for the first 5,000 people. And as with the genetic data, we're not just going to put out the imaging data files. The expert groups in these different imaging areas have been working on automated analysis of the imaging data. And so we will be putting out both the imaging files, but also image-derived variables. So a very large number of image-derived variables from the brain, but also from the heart. Subsequently, body composition ones from the abdomen, etc. But I wanted to focus not so much now on the characterization of the participants, but on the characterization of the health outcomes. Because I think one of the reasons, apart from cost, why geneticists focused on case control studies is that you could identify very well phenotyped cases of disease. In prospective cohorts, the focus is on phenotyping the participants, and then often linking to health records and getting really crude information about their diseases, and perhaps not very specific information about diseases. So the Wellcome Trust and the Medical Research Council, who are supporting UK Biobank, are very keen that we turn these data into really usable information for researchers, and that we are now focusing on the phenotyping of the health outcomes. What will that do? Well, it will enhance the power to detect associations between risk factors and disease outcomes if the people who we think have disease really have disease. The false positive diagnoses have, as we know, the main adverse impact there. Increased specificity of disease classification will allow the detection of specific associations. So, risk factors may be linked to disease subtypes, not to some kind of composite of a disease. And I'll just give one example here. This is breast cancer and whether or not they have hormonal receptors on the breast cancer tissue. If you have hormone receptors, so estrogen or progesterone receptor positive, then associations with parity, age at menarche, age at first birth are seen. Whereas if you don't have, if there are tumors without receptors positivity, then you don't see those associations. So you can imagine if you don't know what kind of breast cancer it is, you'll get a blur. You'll have an average of these two associations. So we can get specific disease diagnoses then we'll get much more specific, more powerful information. And taking that example of hormonal receptors, what about things that we don't know to measure now or to record now? So what we're looking at is whether we can store information about people who are reported to have particular diseases that will allow the subclassification sometime in the future when more is known about how to subclassify. So we've been doing studies in the UK to see whether we can, for people who are diagnosed with cancer, can we identify where their tumour samples are and get hold of their tumour samples and store them so that people can come to them in 5 or 10 years' time with some newly invented marker, test that on the tumours, and then subclassify those. Or to subdivide people with respect to, say, imaging for stroke subtypes. But we have to do this at really enormous scale. These are the kinds of numbers of some exemplar conditions that we estimated will have occurred by 2012, 2017, 2022. And our observed numbers are very similar to these estimates. These are incident outcomes since baseline. There are also, of course, very large numbers of prevalent disease outcomes that were recorded at baseline. So we've got to work out ways in which to develop algorithms to apply other information when we find out that individuals have been reported as having a condition to be able to classify that. So we're looking at a staged approach to doing that. Can we have very cost-effective and scalable approaches to ascertaining suspected cases through linkage to the National Health Service death registers, the cancer registers, the hospital episodes, the primary care records, and then we're using web-based questionnaires directly to the 1/3 participants who have given us their email addresses to ascertain health outcomes that don't get picked up through health record linkage around cognitive decline, around depression, pain, and quality of life. Then looking to see if we can cross-reference to other electronic record systems and disease registries to confirm these cases, and then going even further to review of clinical records, to tumor collections, to imaging databases, to then be able to subclassify the disease cases, so that when a researcher comes to the resource and says, how many people have you got with a lacuna stroke, so ischemic stroke, but with particular characteristics, we can say, these are the numbers with the imaging, they've been classified, here are all the data for those well-characterized stroke cases. So in conclusion, how do you get your hands on this resource? Well, as Neil mentioned, it's available to all. You just have to register on the UK Biobank website and demonstrate that you are a bona fide researcher, which essentially, where are you based? And can we find you? And it's available for all types of health-related research in the public interest. Again, as Neil said, there's no preferential or exclusive access. You only have to pay for the costs of using the resource. So we just have a standard set of costs, a few $100s to submit the application, a few $1,000s to get really as much data as you want to do your research. The costs of setting it up are covered by the funders. And from the funders' point of view, what they consider to be success is imaginative use of the resource by as many different researchers from across the world. They are not looking to recoup the cost of setting it up, which is probably of the order of $200 million at this point. We obviously need to be careful about the access to the biological samples that are depletable, and we are looking at ways of getting additional funding to do tranches of assays. So we're now looking to see whether we can do infectious disease markers and considering other measures in the samples that would be valuable for researchers. And all that you're required to do, as we saw with the UK Believe study, is publish the findings and then return the data so that they go back into the resource and so other researchers can come along and use them. And if someone publishes something from UK Biobank that you don't agree with, you think they've got it wrong, this is the purest form of peer review. Come and get the data and demonstrate they got it wrong, or demonstrate they could have got it righter, or used it in a more imaginative way. So you can apply by going to the website, nice and easy, ukbiobank.ac.uk. On the website, you can look at all the data that's been collected. And we've had a discussion around how this resource could be used for the sorts of research that this community uses. And that UK Biobank is an experiment. It's an experiment in how to make a resource useful for as many different researchers as possible. And we're learning as we go along. There was some anxiety among the funders and the Ethics and Governance Council that was set up to help guide us around having people doing non-specific research on the resource. We argued at the time that that's exactly what they should be doing. They should be exploring it and finding things in it. That anxiety has reduced, and it's been reduced, I think, by the debate about how it can be used by exploratory analyses. And so yesterday we put on the website an agreement from the UK Biobank board that, whereas before we had asked people to put in applications that were quite specific as to what they were going to do, we're now saying that from November, play with it. Use it in the most imaginative ways you can for health-related research. So you have to put in an application. You have to explain roughly what you're going to do. But we're trying to loosen the reins so that it's used as imaginatively as possible. So we hope that you will take up that challenge. Thank you very much.